All right, good afternoon. I was sure that we were going to have a mini lecture from chapter 21, but somehow we almost got through it all, and then I crammed in that last slide. That's recorded, so I will post that so that you can look at that more closely. But I'm glad that we were able to get through 21 without a mini lecture, and now we just have a mini lecture for 20 to go back to. And the reason why I want to have this as a single mini lecture, because there's really one answer to all three questions that we have right here. So these are related to chapter 20. This is mini lecture chapter 20A. And then we're going to move on to chapter 21, which is what we already did. So the three questions, and these all have the same answer in a sense. Why is sucrose such a good storage molecule? How does the mother make milk? And how are foreign molecules cleared from the blood? Those seem like three very different things, but they all have an answer that involves UDP, which I have with a very dramatic animation there, which hopefully will help you remember it. This is what UDP is. You actually may have drawn it earlier when you drew nucleotides, but really it's not used as a chemical functional group in any way. You just use the phosphates for a group transfer reaction, and you can place them onto anything that can attack a phosphate. For example, a glucose can be UDP-ylated because it can be the OH on here on the anomeric carbon of, this is glucose right here. The anomeric carbon attacked the gamma phosphate of a UDP. Actually, I should say it attacked the beta phosphate of a UTP molecule, and it ended up being UDP-ylated. Technically, it's uridynylated, but it's really hard to say, and what you need to know is that UDP is what we're showing when we do this. It doesn't really add chemical functionality at all, anything beyond what just doing a phosphate would, but it adds a tag, a label, that specific uridine structure, and that tags it for anabolism in general. We have a lot of enzymes. There are three enzymes right here that involve UDP and sugars that use the sugars for building. So, question number one is the sugar that is built by sugar plants, including sugar cane, is sucrose. And of course, we don't do sucrose. We eat sucrose, we consume it and break it down. We do not build it. How does sugarcane make sucrose? Well, it involves UDP. And I just want to point out, this is a bit of review that's good again. When you have UDP glucose, the sugarcane will combine it with fructose 6-phosphate. And you see here it's just a synthase that involves that. The synthase is because UDP is being given off. It's not a synthetase because it's not using ATP in the very specific way. It's a synthase in the sense of it's forming a bond between two molecules. It's synthesizing. And so you end up with the phosphorylated, but the original molecule of glucose that gets combined in the special reducing ends, two reducing ends joined together bond, that originally had UDP on it, which tags it for anabolism rather than catabolism. After you do that, they just have a phosphatase that will actually cut off the remaining phosphate that's just on the fructose's 2 carbon, and then you have a nice sucrose molecule. So it all starts with UDP. The reason why sucrose is better than, say, mannose or some other disaccharide that's joined together in the normal way, usually you'd be combining the C1 on the left carbon with a C4 or maybe a C3 on the other carbon. But that's not done. In this case, you actually join it to the fructose 2 carbon, as you see right here. And the reason for that is actually the anomeric carbon on that end. That is more reactive, and it means that it is occasionally forming a ketone in solution. That's the way that these hemiketal bonds are. But when you join it, you actually form the full ketal bond on the fructose side. The full ketal bond is more stable. It doesn't come apart as much, and then you have a more stable molecule. You've taken the two reactive ends and you've joined them together into a less reactive joining molecule. So that's cool. So that is how you make that disaccharide. There's another disaccharide that's very important to all mammals, which is lactose. This is the sugar that babies live on. And so the mother makes the sugar for the baby. There are many components in milk, but one of the main sugar components is lactose. And so lactose is a disaccharide, and the question is, so we don't make sucrose, but we do make lactose. It's a disaccharide that we do make. This one also involves UDP, but it doesn't involve expressing an entirely new enzyme. You don't have a UDP using enzyme technically. Let me show you what you do have. You actually have a modification of another enzyme that is used for just normal kind of glycosylation. Remember that many of your proteins on the surface of the cell, they're all glycosylated, right? So you have sugar transferases that will transfer the different sugars onto the growing glycoproteins that will then be put on the surface of the cell. These are basically little factory workers that are putting on the different sugar units that will eventually coat and protect the sugar molecules that will be joined to the surface proteins. So for example, sometimes you want to put a galactose onto an acetylglucosamine group, and everyone has a particular enzyme that forms this bond. This happens in non-lactating tissues that need glycoproteins, and it's just a very ordinary sort of enzyme to have around. The thing about it is it works on UDP. Like sucrose, it will join this bond together. The end product in this case is not necessarily a disaccharide, but an oligosaccharide that will be joined to a glycoprotein. The thing is that lactating mammary glands will actually need to make this lactose for the baby. And what they do is they join galactose instead of to glucosamine to glucose itself. And then they take that and they give it to the baby, right? So they have a lactose synthase is the name of the complex that makes this. But it's a complex, not an entire enzyme by itself. It turns out that lactose synthase is composed of two chains, one of which is alpha-lactalbumin, which is a shorter chain, which modifies the specificity of the ordinary galactosyltransferase. This is more complicated than just making a protein, but it's also easier for the body to do than to make a whole protein, and the effect is the same. What it does is it changes the galactosyltransferase so that instead of binding glucosamine, it binds glucose. And then it can go on and it can make a bunch of the lactose for the baby. It's kind of cool how this is a less drastic change. It's making half a protein that changes the function of an existing protein rather than making a whole new protein. It's probably more efficient when you look at it that way. It's a little more complicated to remember. But the alpha-lactalbumin is a gene expressed after childbirth that will actually change the specificity of the enzyme. So this brings to the question, for organisms with XY chromosomes, can XY chromosome organisms lactate? And the answer is sure. Actually, they can. It's just a matter of getting a gene expressed, and the gene is not even a full protein. It is a protein that modifies another protein. I don't know exactly how big it is, but it's probably simpler than a big protein, so it's probably easier to turn on and off. Evolution certainly figured out a way to do that. And so basically the hormones, the same hormones that will cause expression of alpha-lactalbumin in XX organisms will result in the expression of alpha-lactalbumin in XY organisms. I wonder, as a scientist, then could you just cut to the chase and maybe inject lactalbumin, and would that work? There's probably some other things that the hormones cause. So this is probably a hormonal thing that can do so that biological men can lactate. So there are numerous conditions under which injected hormones have produced it, and sometimes it's an expected effect, and sometimes it's an unwanted effect. And so hormones do this. It's the way hormones work. And hormones are actually a handle that we have that we can use to manipulate chemistry. So there was a study where there were male and female cancer patients who were treated with estrogen. A lot of cancer treatments do involve hormones. And they, both the men and women, secreted milk when they were injected with prolactin, which I believe is another hormone. I'd have to look it up for sure. And there are tranquilizers that actually affect the hypothalamus. So these don't directly involve the hormones. They involve the brain, which makes the hormones. And that will actually cause milk to be expressed. And even sometimes if there is surgery that somehow like rewires the nerves related to the suckling reflex, that can actually cause the expression of this. We don't know exactly all the dots that are connected, but there are lots of ways to express the lactating genes, and we can figure out what those are, and then we can manipulate them for certain cases. And it's important as a doctor to be aware of how powerful hormones are. So the final topic is one that seems entirely different. It's how are foreign molecules cleared from the blood. This itself also involves UDP. So it turns out that this involves a different kind of UDP elated. We've talked about how UDP glucose is produced and used for anabolism. Well, that can also be dehydrogenated to UDP glucuronate. This is one of the ones that was mentioned back in the chapter on sugars, but it's just glucose where the carboxylic acid has been produced, I believe, at the C6 carbon, rather than having a hydroxyl. It's oxidized, and that's all that you have to have: an enzyme that binds the UDP glucose. It oxidizes that one carbon, and then you have an acid sugar instead of a regular sugar. It turns out that UDP glucuronate is used for two purposes, which is for tagging drugs to be removed by the kidneys, and then it's also the source of vitamin C in the animals that can make it, which is not us, by the way. Most mammals can actually make vitamin C, but we had enough in our diet that evolution just decided it was needless and threw away that gene because it decided that we would be eating enough and we survived well enough until we started going on long sea voyages, and then it turns out, oh, we need vitamin C in our diet. So they figured that out pretty fast. So that's the whole vitamin C story. Both of these stories involve UDP. And there's UDP glucose. So UDP glucose, you actually oxidize it twice. You take off 4 electrons, 2 electron pairs, to make UDP glucuronate. And you see right there is what I call the periscope carbon. It's the C6 that sticks up from the rest of the ring. UDP will turn that into a COO. And then you can use that whenever you need a more highly charged sugar. By the way, sometimes you need more highly charged sugar for the glycosaminoglycans, which are mentioned in Chapter 6. I think it's Chapter 6. It's Chapter 7, the sugar chapter, whichever one that is. And so if you just need a COO on a sugar, you can get it this way. There's a dehydrogenase that produces it from normal old UDP glucose. You can also use it for other purposes. You can use it for glucuronidation, because it's glucuronate being put onto something. Or you can just use the glucuronate itself as a scaffold to make another small molecule. The small molecule that it makes vitamin C. And the whole thing about this is most mammals have this. We don't. And also guinea pigs, for whatever reason, they must eat enough citrus in the wild that they don't need it either. And that's why guinea pigs were used for this. You don't use vitamin C, the glucuronate derivative that is vitamin C. You don't use it for many things, but one of the things you use it for is for high-powered oxidations. And so you use it for hydroxylating the proline in collagen. If you don't get vitamin C and you're a guinea pig or a Homo sapiens, if you don't get that vitamin C, then you don't have good collagen anymore. And that means you start bleeding from the gums, you start having joint problems, all the places where collagen holds you together. You don't have that hydroxyl group that's really important. The hydroxyproline is central to collagen's function. If you don't have that, you don't have good collagen. So Glucuronate is the other part of that. And a glucuronate tag will actually be used for other purposes. This is a purpose for just if you have lipids that are hanging around the body, you want to get rid of it. This is a lipid that's produced in cigarette smoke. When you inhale cigarette smoke, you get this combustion product in your body, and it's fat soluble, so it's hard to get rid of. In fact, it really helps to make it more water soluble to be able to get rid of it. It's only got one little place where you can even do any chemistry with it, but it turns out that little place is enough for a transferase to put a very soluble molecule onto this insoluble molecule and to make it more soluble so that then the kidneys can flush it out of the system. This is really cool biochemistry. This makes it more water soluble, so this ends up being able to dissolve this cigarette smoke derivative. Okay? So this is also a carcinogen. It's not just any old lipid. It actually causes cancer. And so it's a good idea for your body to get rid of it. Also, honestly, I wonder when I see any uncontrolled people who tell me that smoking pot is just like smoking, is much healthier than smoking tobacco. Maybe, but if you're smoking it directly, you still are getting these uncontrolled combustion products. And the uncontrolled combustion products can look just like that. So I'm not sure that, now there are ways to deal with it so that you don't get uncontrolled combustion products in your system. But I won't talk about those ways. I'm just going to say combustion is combustion. And so you're probably getting this stuff in your system if you're smoking, however you're smoking, as long as you're inhaling smoke. But so this enzyme basically just has a really hydrophobic site that goes around and clears up these hydrophobic pieces of crud from your body. It also does this with AZT, the HIV drug, and the sedative phenobarbital. Those are both very hydrophobic, or whatever they are, they stick to this enzyme. And they both are made more soluble by a glucuronate tag. And it starts with a UDP joined molecule. So increased exposure to any of these will cause your body to detect there's more crud in my body, so I better make more of this transferase. And so your body is trying to react when you are putting toxins into it. So just make sure that those toxins are something that your body can handle. This is how tolerance is actually evolved, and how when you smoke a lot of smoke, your body does adapt. Maybe you can measure transferase levels to see if someone's a chronic smoker or not. There's one final use for UDP. Bacterial cell walls are usually talked about in the membrane chapter, and they're actually made of linked carbohydrates. So the bacteria, when it's building its cell wall, it will be building with a sugar, and therefore it's going to be using UDP. It's the very first step that brings the very first sugar in when a cell wall is built. UDP sugars are also used for building cellulose. So you see, it's a pretty good rule. If there's a sugar involved in building things, then it's probably got a UDP-linked precursor. And I just want to remind you, penicillin, one of the most important drugs in history, will actually be involved in this process. It's not involved with the UDP step. It's involved with the last step of assembling this same structure. It's actually a transpeptidase that will join amide bonds together. But you can talk about that, but that's not this chapter. We're talking about carbohydrate biosynthesis. I just want to mention that it's involved with that. All right, so that finishes up chapter 20. We're done with chapter 21. We've got one more chapter and then test 2. So here's the homework. This is all online. And I will see you Monday. Or if you're watching this after Monday, I will see you Wednesday in class.